My name is Mariam Boules. I'm a senior biology major at Tennessee State University, and this academic year I was working with Dr. Margaret Whalen as well as PhD student Sharietta Brown, and we were examining the role of MAP kinases as well as NFκB to look at Tributyltin-stimulated induces increases in interleukin-1 beta production in lymphocytes. Interleukin-1 beta is a really great cytokine, so it promotes cell growth, tissue repair, and inflammatory response. However, increased production of interleukin-1 beta can actually lead to different autoinflammatory diseases as well as certain cancers. Disruption to the cytokine can be triggered by a stimuli, and which can be found in the environment. This stimuli happens to be TBT. TBT is a highly toxic biocide mainly used in the anti-fouling paint industry to basically prevent the growth of organisms on the hull of the ships. So basically the goal is to help the ships move smoothly in the sea without any disruption. As a result, the compound basically over time breaks down, goes to the sea organisms such as fish, and we are great consumers of fish, so we end up consuming fish, and as a result, research have found that the compound has been found in our blood samples with concentration as high as 261 nanomoles. As a stimuli, our goal was basically to examine the different pathway that TBT utilizes to increase interleukin-1 beta production. And based on previous studies, it has been shown that TBT utilizes ERK1/2 and P38 pathway to increase interleukin-1 beta secretion from monocyte-depleted peripheral blood mononuclear cells. The goal for this project is to basically examine the role of MAP kinases, which are ERK1/2, P38, and JNK, as well as NFκB, to examine TBT-induced increased production of interleukin-1 beta. And we were able to accomplish this by obtaining our blood filters from the Red Cross at Nashville, in Nashville, Tennessee, and to measure, and we also used ELISA assays to measure the secretion levels of the protein, as well as we used Western blots to basically measure the intracellular levels of the protein. Results showed that P38 pathway and ERK1/2 were actually utilized by TBT to increase interleukin-1 beta production. However, JNK and transcription factor and NFκB were not utilized by TBT to increase interleukin-1 beta production. Finally, our data suggests that when we are exposed to TBT, we are prone to inflammatory diseases when levels of interleukin-1 beta are increased in the activation of MAP kinases. Thank you for stopping by. I will leave my contact information at the end of this video if you have any questions. Thank you.